Thank you, Kayla. Good afternoon, good morning, good evening, everyone, depending on where you are joining us from. Thank you for taking the time to join this GFRA webinar series. We are very excited today to host the very first GFRA African chapter webinar. For those of you who do not know, the GFRA African chapter is a sub-working group of the GFRA. It was established in 2023 at the GFRA scientific meeting in Kampala in Uganda. So how very fitting that our very first African chapter webinar is from our executive committee member of the African chapter, Dr. Susan Kerfua, who is the regional leader for East Africa. So we're very excited to have Susan with us today. And thank you so much, Susan, for accepting the invitation. And it is my privilege to introduce Dr. Kerfua to you. So Dr. Susan Kerfua is a scientist at the National Agricultural Research Organization of Uganda, based at the Livestock Resources Research Institute, where she focuses on viral diseases, particularly foot-and-mouth disease virus. She holds a PhD in life sciences from the Nelson Mandela African Institution of Science and Technology in Arusha, Tanzania, as well as a Master of Science in Molecular Biology and a bachelor's degree in biomedical laboratory technology from Makerere University, Uganda. She is a dedicated researcher with extensive expertise in molecular epidemiology and virology. Dr. Kerfua has made contributions to the understanding and management of infectious livestock and zoonotic diseases in Uganda and East Africa. Beyond her core responsibilities, she is actively involved in the Global Foot and Mouth Disease Research Alliance's African chapter and serves also as the chairperson of the Uganda National Animal Health Laboratory Network. She has also undertaken short-term consultancies and played key roles in internationally funded projects, demonstrating her leadership and technical expertise. So, Susan, welcome again, and we look forward to your presentation. Thank you very much. Over to you.
Oh, thank you so much, Melanie and Kayla. Very honored and privileged to be presenting at the GFRA webinar series. My name is Susan Kerfua, working at the National Agricultural Research Organization, particularly with the National Livestock Resources Research Institute. First of all, I would like to thank everybody who has taken up their time to be here to attend this. Good morning, good afternoon, good evening, wherever you are. Thank you so much for joining us for this webinar series. Together with my colleagues Cosmos Openja and Vincent Karagendo, we are happy and honored to be presenting on this kind of research work. I'll be moving on. Our title is Current Situation and Research on Foot and Mouth Disease in East Africa. Particularly, we're looking at the years between 2000 and 2024. This is about 2 decades. I'll be referring it to about 2 decades. So thank you so much for joining us. Maybe we'll move to the next slide. Yes, so we very well know that foot and mouth disease is a highly contagious disease that affects all cloven hoofed animals, which includes livestock species as well as domestic species. The signs are very obvious, particularly in cattle, where we find there's drooling and salivation, lesions on the mouth, the hooves, the udders and femurs, but also we see cases of deaths in calves. But it's important to note that there are some wildlife species that may not be able to actually show obvious signs of foot and mouth disease, like the small ruminants, that is the sheep and the goats, and yet they play a very important role in the epidemiology of this disease. Now, foot and mouth disease has very devastating economic consequences, especially to continents and countries where the disease is endemic. This is reflected by Knight Jones et al. Quite a number of people have done this research. So it's very devastating economically, but also we know that foot and mouth disease has implications to food security, because usually when there's an outbreak, we find that there's a ban on sales of beef or meats for that matter. And also we find that sometimes there's a ban on sales of milk or even movement of milk. So we find that the vulnerable people who actually need this nutrition cannot be able to get it because there's a ban on sales of these. We also know that the social aspects of society can also be affected by foot and mouth disease outbreaks. For example, societies or cultural practices that require live animals, for maybe wedding ceremonies or even rituals, certain rituals and cultural issues. Sometimes they have to use live animals, and so when there's an outbreak, we find that some of these they cannot practice some of their cultural norms. So foot and mouth disease is very, very important and very relevant to the society. So the pictures just show classical signs of the disease that I've been talking about, the lesions on the tongue and also in the mouth or in the oral cavity. Next slide, please. So this is just a map that I borrowed from the World Reference Lab. We can see that there are some continents that are quite endemic for foot and mouth disease, which includes Africa. You can see the sub-Saharan Africa is quite endemic, and Asia rather, and Asia is also quite endemic. But we realize that there are some countries that actually have sporadic outbreaks. For example, most recently we had also Hungary having a sporadic outbreak in March. So there's that occasional, incurrence of outbreaks in some of those countries that may be free without vaccination. So you realize that the countries that are endemic are trying to actually ensure that they get free of foot and mouth disease without vaccination or with vaccination. But also the countries that are free of foot and mouth disease are ensuring that they close their borders off, to this virus. So there's that, that struggle for both, for all kinds of countries to ensure that they get rid of this virus. For this talk, I'll particularly center on East Africa, which you'll see that for some data sets, I will include the larger East Africa, which will include Rwanda, Burundi, Uganda, Kenya, Tanzania, South Sudan, and the DRC. But also for other data sets, I'll zero down to specifically Uganda, Kenya, and Tanzania. Next slide, please. So foot and mouth disease exists in 7 serotypes. We have OA, C, Asia 1, SAT1, SAT2, and SAT3. And it's very interesting that this disease kind of has a geographical distribution, and that's why we talk about the pools. So for East Africa, we have pool 4, which consists of serotypes OA, SAT1, SAT2, and SAT3. So of course, still the red still shows the countries or the areas that are endemic for foot and mouth disease. So I'll be zeroing down on East Africa, which is for pool 4. Thank you. Next slide. So when we're starting to do this work, what were we seeking? We were seeking to investigate the distribution patterns of foot and mouth disease, virus serotypes and tophotypes in East Africa. And we started to do this through a thorough analysis of, research data and, works that people have done. But also we decided to look at some of the virus samples that have been submitted to the World Reference Lab of Foot and Mouth Disease website. And we also sought to assess the antigenic matches that are between most of the FMD vaccine strains that are being used and also the circulating virus strains in East Africa. So these were just some random ideas that we thought of. Were like, oh, okay, why don't we, find out what's happening in East Africa based on information, retrospective data, what people have actually done, and come up with some information and data on this. Next slide, please. So what we did was to do a systematic review considering available data from research articles and from the World Reference Lab FMD website. And like I explained before, we're looking at the years between 2000 and 2024, specifically looking at the Eastern African countries. Here I put Uganda, Tanzania, and Kenya, but you'll see that some data will include other East African countries. Also, we looked at information in regard to antigenic vaccine matching for this work. Next slide, please. For what did we find out? For regarding what we found out is that if we looked at the sample submission to the World Reference Lab for foot and mouth disease per country, we found that Kenya had the highest number of samples submitted, followed by Uganda and then Tanzania, whereas the other countries were a bit lagging. That is Burundi and Rwanda were lagging in terms of sample submission. When we look at the samples that were viable, samples that were able to give some viral growth, we find that the samples had some issues, because when we look at Kenya, we see about 150 samples not being able to give viral growth. and in Uganda 158 samples. Same story goes for Tanzania, that there were some samples that were not able to give viral growth, and therefore there was no significant information that could be gotten out of the samples that were submitted. So if you look at all the countries, most of them actually had samples that did not have any viral growth. And for Rwanda, it was not very good because, I mean, over most of the samples, about 90% of the samples did not have any viral growth. Next slide, please. And this leads us to like reflections. We need to start thinking about this. First of all, it's important to note that 99% of the samples were from cattle, and then we had about maybe 1% of the samples being from buffaloes. That is the African buffalo. So, like I said before, there are implications to this because we have sheep and goats that can actually be able can be actually be able to spread the virus even if they're not showing any clinical signs. So what does this mean? Does it mean that probably maybe if there's an outbreak, we might have to also collect some samples from the sheep and goats and figure out if they are positive or negative for foot and mouth disease? Because if there's an outbreak on a locality that has cattle and sheep and goats, we need to ask ourselves questions like, oh, what's happening in the sheep and goat population that is next to or near the cattle population? So this also leads us to start thinking about how can we actually design our surveillance plans, our surveillance tools to ensure that also we are able to capture the sheep and goats in our surveillance programs. And also we need to start thinking, why was it that most of the samples did not have most of all of the countries actually did not have samples with viral growth, and maybe it's cold chain issues. This is something that we know is common in Africa. We have this challenge of when we go out there in the field, sometimes we're going long distances, even over 100 kilometers to collect virus. So how are we able to maintain this cold chain to ensure that the samples reach the lab in the right condition, and we are able to get viable data out of it? But also think about it. Maybe it's about poor sample collection. Are we actually picking the right samples? Uh, you know, do we know that, maybe if we pick epithelial tissue that's fresh, that's what we'll be able to get the virus, or do we just go out there and pick whatever we have to? So this also brings in implications of training. How frequently can we be able to ensure that this training is done for labs that especially participate in foot and mouth disease research? Also thinking about the timing of sample collection, what's the viable window in which we can be able to get as much of the virus as possible? So all this comes into training, as well as packaging of samples for shipment. When the samples are being packaged for shipment, wherever it could be in country, it could be outside the country. For this instance, it's the World Reference Lab. Is it being done the right way? And still goes back to training. How many people are actually trained in good sample shipment to ensure that the samples are as viable as they can possibly be? There could be other reflections that we can think about. I'll be happy to take them on maybe in the questions and comments. So there's still others that we can actually think about. Next slide, please. Yes, so we looked at using all this data and information. We looked at the serotype diversity in East Africa. This is over the last 24 years. And we see that, wow, there's quite a lot of serotype diversity in Africa for most of the countries. If we look at Uganda, for example, over here, we see that within these 24 years, we had 5 serotypes in circulation within these 24 years, and this includes that 3 having been gotten in a calf. And then when we look at Kenya over here, light purple, we see that they also had 5 serotypes in circulation. This is 5 out of 7 serotypes in circulation, which included also serotype C, which for now is extinct. But then in 2004, they had serotype C. When we look at Tanzania and Sudan, they have the same number of serotypes. And that is serotype OA, SAT1, and SAT2. So these are the serotypes that we find in Eastern Africa. So we see that this is a high diversity. But also to note, we might find that actually there are even more serotypes in these other countries, that is Burundi, Rwanda, and DRC, that than is being recorded, because we know that there was less sample submission, so there could be instances of that, of the fact that maybe samples were not submitted, samples were not collected, and therefore we were not able to see maybe much of the diversity in those other countries. Moving on, please. Yeah, let's take a look at Kenya. Looking at the years between 2000 and 2011, we can see that the most prevalent serotype was serotype O. And within serotype O, we see topotype EA2 in circulation between 2000 and 2011. And we also see that there is serotype EA1 in circulation in Kenya about that time, and EA4 as well in circulation about that time. This is a bit different. If you look at the years between 2012 and 2023, we see that somewhat we don't see topotype EA1 serotype O. We don't see serotype serotype O, EF4 in circulation anymore. But then we have a new serotype showing face, topotype showing face, and that is serotype O, EA3 showing face. So we already see that there's already some kind of change in terms of topotype distribution. And this can actually have a lot of implications when it comes to intervention by vaccination. Maybe just to say is that the foot and mouth disease serotypes are not cross-protective. And even sometimes in instances, we find that the topotypes, there is no cross-protection between topotypes. So all this has implications on the type of vaccines that are used, the strains of the vaccines that are used for these different viruses. So that is about all. To notice that is that it's the most prevalent serotype. It was the most prevalent serotype in Kenya at the time. But also we see that A, serotype A was also quite prevalent, and same thing with SAT2, and SAT1 also was coming up. But also we see that between 2000 and 2011, serotype C, the Africa topotype, had been detected in Kenya, but now it is not there anymore. So we see a high diversity in the first decade, and then a much lower diversity of serotypes in the last decade. Next slide, please. Yeah, let's look at the case of Tanzania. Tanzania, in the past decade, we see maybe a low diversity, less by one serotype. But in the second decade, that is between 2011 and 2021, we see 4 out of the 7 serotypes in circulation in Tanzania. And well, it's just to say, not to say that, oh, probably in between 2000 and 2010, there were no, the other serotypes maybe were not there, but it still comes down to the same thing of, are we being able to track outbreaks the way we should? Or is there an issue of reporting of outbreaks such that certain samples are missed out, and then we cannot get the definite diagnosis or the definite serotypes and topotypes that are in circulation? So anyway, we still see that in the last decade, that is 2011 to 2021, we see that there's 4 serotypes in circulation. That is all. We have A, we have SAT2, and then we have SAT1. And also it's important to note that the O topotype is still the same, which is same as for Uganda, that is EA2. It's still the same as for Uganda. And note that the one for Kenya, we had EA2, we had EA1, and we had EA4. So all this has implications when we're coming together to talk about foot and mouth disease control. Next slide, please. Yes, coming back to Uganda. So still same story. We still have a lot of serotype diversity and even topotype diversity. In the first decade, we still see serotype O, EA2, featuring quite significantly, most prevalent as usual. And then we see A, and then we see SAT2 and SAT1. So all these are there in the previous decade. Now, in the latest decade, that is between 2011 and 2013, we see an inclusion of SAT3, which is over here. We see an inclusion of SAT3, which was detected in 2013, as well as we see, if you look at serotype O, we see actually an inclusion of East African 4 topotype. And yes, this is interesting because it's something new. Because previously it was not in Uganda, but now it's something new, and probably it would have come from maybe the neighboring countries. But of course, the years between 2011 and 2023 presents a higher diversity of circulating strains, and I mean circulating topotypes. And of course, like I said before, this has great implications when we're going to look at intervention through vaccination. Next slide, please. Yes, so looking at the reflections from this, what do we get from this? First, we've seen that the highest number of submissions were coming from Kenya and Uganda, and this is in tandem with some publication that was recently put out by Renault et al. They showed that actually Kenya and Uganda were amongst, including Tanzania, were amongst the highest the highest submitters of samples to the World Reference Lab, which is a good thing. So the only issue is that some of the samples are not as good. They're not submitted as good, so we cannot be able to get the definite results that are required. We also see that foot and mouth disease is endemic in East Africa. At least every year, there's an outbreak in each country, every single year. So this is, and we're talking about since the 1950s, because most of these countries had their first outbreaks in the 1950s. So every single year since the 1950s, there's been outbreaks recorded. So we need to ask ourselves, what next from here? Are we going to go through another decade like this, or what interventions do we have to think about? We see that this is the region that has had the highest diversity of foot and mouth disease serotypes, considering that the region had all of the six serotypes out of the seven serotypes. The only serotype that we did not have was Asia 1. So, 6 out of 7 serotypes, that has a lot of implications when it comes to disease control, has a lot of implications when it comes to diagnosis. It has a lot of implications when it comes to surveillance and controls. When we look at Kenya, yes, serotype C stood out. That was in the first decade, which was it was detected in the year 2003, but now has disappeared. So the last time it was detected was in 2004. Uh, Serotypes O, as we have already seen, is the most common serotype, and I think this is a global phenomenon. In most of the countries, serotype O is the one that is dominating. But like I said before, we realize that there's still different topotypes of this serotype in each of these different countries. Particularly Kenya had the highest diversity of serotype O topotypes. And yes, Uganda also had an incursion of the EA4 topotype, and we'll be seeing later that, of course, this is going to have implications in terms of control using vaccination. We also see the challenge of some samples not being genotyped, because if you look through the graphs, you realize that we had maybe serotype O, NM meant not mentioned because it was not genotyped. So we have issues of samples not being genotyped, and this is a challenge. It's a challenge because when we get into vaccine matching, it's difficult to actually know which strain of the virus this is, and therefore difficult to ascertain even which vaccine strain to consider for administration. Um, so that is a big challenge, and as a region, we need to figure out how we would go about it. Then, of course, the incursions of certain serotypes and certain topotypes is also a factor that we see that stood out, and all this, like we said before, is very important when it comes to interventions. Of course, most of the samples were from cattle. There were I think from our work, none of the samples did we get from even the sheep and goats and pigs. None of the samples. Yeah, there was one sample from pigs. Yes, one sample from pigs, but otherwise, most of the samples were from cattle, and a very few of the samples were from buffalo. So yes, like I said before, we need to think about how we can do a very good sampling. I mean, not something, a very good strategy in terms of surveillance. Yes, thank you. Next slide. Yes, now talking about vaccine strain matching, forgive the repeat of the word vaccine strain matching. Yes, we thought to also look at what is in terms of vaccine strain matching. So we looked at Uganda, Kenya, and Tanzania, and we realized that actually Kenya had submitted a lot of samples for vaccine matching, and these results are just showing for serotype O, but the other serotype, this is just one of the examples that we're putting out there, but the other serotype strains that underwent vaccine strain matching. So when we look at Uganda, Tanzania, and Kenya, Kenya had the most samples that were submitted for vaccine matching. Um, any figure that is anything that you see that is below 0.3 means that the vaccine strain, maybe just to show you, these are the vaccine strains. I don't know if you can see that. Um, Below here are the vaccine strains that were used to be tested against the circulating strains of the virus. For example, circulating strains would be gotten from 2019, 2020, and 2022. Anything above 0.3 would be said to be protective, whereas those that are below 0.3, there would be some question marks about it. So we can see that actually the strains can be different, can rather exhibit different R1 values. So these are R1 values. R1 values below 0.3 and may not be protective compared to R1 values above 0.3. So you may find that actually you have different strains of the virus throughout the different years, and they may behave differently actually towards the vaccine strain. Next slide, please. So, for example, we got all this data from these reports, the FMD vaccine matching strain differentiation reports. That's where we got all this data from. So, this is just an example of a report from which we got this data. This is a report for Kenya, and we can see that the strains between this is a strain from 2021. I don't know if my pointer can be seen. Okay, this is a strain from the first column. Under A is a strain from 2021, and we can see the results. The first figures before the comma show the R1 value. If the R1 value is below 0.3, it means that the vaccine strain may not be protective against that circulating strain. So if you see over here, we have a lot of figures that are below R1 value 0.3. So it means that, okay, this vaccine may not be able to work. I'm using may because, I mean, this is something that is done in the lab, but we don't know what could happen out there in the field. Next slide, please. Yes, so when we analyze this data for the vaccine strain matching data, like I mentioned before, Kenya had the most number of samples that were analyzed for vaccine strain matching. And going back to what I told you, an R1 value of less than 0.3 may mean that that vaccine strain is not as protective. And an R1 value of more than 0.3 means that that vaccine strain may be protective. So if we look at these graphs, we shall see that for Kenya, serotype O strains, for the vaccine strains that were tested against serotype O circulating strains, most of them were protective, seem to be protective. Not so with the rest of the other strains. For example, if you look at serotype O strains, when you look at SAT1 strains, SAT2 strains, the results are not as good. And of course, like we said before, this has a lot of implications on what vaccines have to be administered, where, and when. If we look at Tanzania, Tanzania and Uganda had very low, submissions for vaccine matching. And if we look at Tanzania, we see a kind of mixed story because over here we see that there's that 50-50% chance of whether the vaccine strain can be protective or not. So over here we see that for all A SAT1, there's a 50-50 thing. Some of the vaccine strains were not protective, and some of them were protective for the samples that were submitted. But when we look at SAT2 strains, yes, for SAT2 strains, most of the strains that were tested against the circulating strains were protective. If we look at Uganda, Uganda did not submit many samples and only submitted samples for serotype O and serotype A. So we do not have results for SAT1 and SAT2. This, of course, still has implications because if you're considering testing for vaccine matching, you have to submit all the samples for viruses that are circulating within the country. Remember that in Uganda we had serotypes O, A, SAT1, SAT2, but now the submissions are only for serotypes or an A. Well, nevertheless, we see that serotype O strains reacted better. They reacted, rather, the serotype O strains of the vaccines by giving good vaccine matches compared to serotype A. So we see that most of the values for serotype A were quite low. They were lower than zero point 0.3. Next slide, please. So what are our reflections on this? Yes, we've already said that Kenya had the most samples assessed for vaccine matching, which is good. This is very important for our country because it helps our country to know, what vaccines to actually administer in the face of an outbreak. So this was good for Kenya, but Uganda, we had the least number of samples that were assessed for this same taste. And yes, I will still say that it has great implications when it comes to administering vaccines because it means that we shall just administer vaccines blindly without knowing, without any assessment. So the vaccines will just be administered blindly. We will not know if they're protective or not. So it's very important that countries actually do this vaccine matching. We see that Uganda had the least number of strains assessed. Yes, I've already discussed that. And we see that for Kenya and Uganda, all, all vaccine strains performed best. For Kenya, probably yes. For Kenya and Uganda, probably it's because of the fact that the vaccines that were used that time were the strain from Kenya, so probably the strain was quite close to most of the vaccine strains, I mean, the virus strains that were in circulation. So that could maybe explain that. And we see that for Tanzania, such two vaccine strains performed best compared to the rest of the others. But also, it brings us to the point of our countries actually following up on if the vaccines that they're using work. Our countries performing vaccine evaluation studies, um, or are countries just administering vaccine blindly? Because vaccines are quite expensive, and so it's imperative on a country that they should be able to follow up if a vaccine is performing the way it is supposed to perform, because then the countries can be able to have discussions with the vaccine producers and say, oh, maybe we need to tweak this, we need to change something in this because it's not working the best way it should. So all these are things that countries have to think about, have these reflections, particularly on vaccine evaluation studies. Please recall that these sample submissions, what we're looking at, were from the year 2000 to 2004, but we've had very few vaccine evaluation studies that have been carried out in each of these countries. Next slide, please. Yes, so just to conclude, these are just conclusive thoughts. Um, we need to ask ourselves, how are the epidemiological patterns helpful to each of these countries, or even to the region as a whole? How can this be helpful in terms of temporal and spatial distribution? How can we be able to make use of this data to infer foot and mouth disease control? For example, why are certain serotypes persistent in certain places? Why are certain topotypes coming in to certain places? So these are some of the questions we need to ask ourselves. And when is the time that these serotypes or topotypes come into particular places? Is there some kind of pattern? For example, do we see serotype or having some kind of pattern in regard to temporal distribution or on regard to spatial distribution? And then what can we do about it? So I think this is, we can make the most of such kind of information to help in the control of foot and mouth disease. Then the next thought is, how can we deepen collaborations, regional collaborations, and ensure that we synergize, we have some kind of synergy, be able to utilize make the most of the resources in each of these countries, the East African region. How can we be able to collaborate in terms of training, in terms of diagnosis, in terms of research? Do we have any collaborative projects? These are things that we have to think about, because we have the same, and we have the same enemy. So why are we doing things apart from each other? Why don't we come together, have a concerted effort to see to it that we try to put this enemy down? Then I also want to talk about communication between industry, researchers, and policymakers. When do we ever sit down? For example, when do the researchers ever sit down to actually tell the industry, the vaccine producers, that we think this vaccine has a problem? This vaccine is not working for us. Do you think you can make it better? Do you think we can tweak something about it? When can we have these conversations between researchers, industry, and policymakers? And the policymakers are also very important, because they need to know, which policies to put out there in regard to even a vaccination schedule, in regards to vaccination programs. So we need to have more talks together, instead of working in silos, working in isolations, because the researchers usually stay in our labs, and we're just in there locked up, trying to come up with something, and we are just in our own little, bubble. So when do we step out and meet industry and have discussions on how to improve vaccines? When do we meet policymakers to have discussions on what they think about the work that we're doing? Then looking at stakeholders working together to control foot and mouth disease, veterinarians, farmers, we have also the leaders, the local leaders, actually, very, very important people. We have the people along the meat value chain, the milk value chain. We have all these people. Uh, when are we able to engage them, as researchers, and hear from them? For example, we know that all this information that I've presented is not has limitations, because we know that there are outbreaks that were not even reported. There are samples that were not even picked. So you find that this information that I've presented is just a portion of the real thing, of what actually transpired. So how can we engage farmers so that they can be able to report an outbreak and not fear for the consequences of reporting this outbreak? Because in Uganda, we've had quite a number of outbreaks, but you just hear that there was an outbreak like 3 weeks ago. After, when you go to the field, you cannot be able to collect the kind of sample that you want to collect, because the farmers are fearful that when they report an outbreak, the quarantines are going to be put up, and then their lives are going to be miserable. So how can we be able to engage farmers to also be able to help us to be able to collect the samples in time, to be able to give them the feedback that they need? So maybe we need to bridge these gaps in terms of communication and in terms of working together. So next slide, please. Yes, so I'd like to acknowledge all these different entities that have been very instrumental. I'd like to thank NARO, where I work. I want to thank the Ministry of Agriculture, Animal Industry and Fisheries, Uganda. I'd like to thank GFRA for giving me this opportunity to share this work. I'd like to thank the USDA, where we are collaborating together on a number of projects. I want to thank CRDF Global for facilitating this World Reference Lab Foot and Mouth Disease. You make such a big difference, and we want to thank you for the work that you do. I'd like to thank GFRAC, the African chapter, and to all our colleagues who have joined from GFRAC. Proud of you. Thank you for joining. I also would like to thank the FMD scientists that are tirelessly working, on doing work on foot and mouth disease. Thank you so much for all the work that you do. All the scientists from East Africa, the popular ones, Kasanga, Mwine, we have Dikusoka, we have quite a number of these scientists, and we're very grateful for the work that you've done. We have the veterinary and lab professionals who work tirelessly, to do a lot of work. And also we have the farmers. We cannot forget the farmers. And I'd like to thank this audience. Thank you so much for sparing your time to listen to me. Thank you.
Oh, thank you, Susan. A very, very nice presentation for giving us this lovely situation analysis of FMD in East Africa. Really a nice talk to start us off with the GFRA webinar series. Just a reminder to our participants, please, that you will see there's a Q&A box. Kindly pop your questions in there and we will try and answer all of your questions before the end of this hour. So we have some questions from Aldo Decker. Susan, thank you for your overview. Very interesting. I was wondering, you showed quite a number of samples that were not possible to cultivate, but were these samples positive by RT-PCR, or could the clinical signs also be caused by other infections or trauma? So that was one question.
So should I answer each or
I
wait
think
for a
so.
couple?
Then we go on to the next. Yes, thanks.
Okay. Um, so yes, this is a very good question because you find that in the analysis report they will say there was no viral growth. You find that most of these, if you look at the data from the countries that are sending these samples, most of the data says that they were positive by RT-PCR. Yes, which is fair enough. But now when they reach the world reference lab of foot and mouth disease, they don't even have they're not even positive for PCR. So that means there could have been an issue, during the transportation or whatever it is, processing, whatever it is. So yes, these are some of the insights that we need to discuss. Could it be that the wrong samples were collected? Like Aldo, you're suggesting, could it be that the wrong samples were collected and all that? So these are the issues that we have to really delve deeply into. Thank you for your question, Aldo.
Just to follow up on that, what is the diagnostic capacity of the East African lab? So are you only able to do the molecular plot, serology wise? Yeah,
Oh,
if you can
yes, that's a
elaborate.
very good, yeah, it's a very good question. Yes, we mostly do the serological analysis and also PCR. We can do conventional PCR and we can also do real time PCR. When it comes to virus culture, we have not yet in Uganda. We've not yet set up that system, but I know Kenya has set up that system. Tanzania, they're doing a lot of sequencing. They can be able to do virus sequencing. So, so those are the things that we need to sit down together as a region and then
Mm,
ask ourselves, how can we
mm,
make the most of the of the materials or the resources
mm.
in each of these countries? Yes.
Okay, thank you. Okay, next question from Aldo. I think he's just correcting your interpretation of the R1 values. He says that above zero point 0.3, there is no significant difference between vaccine strain and outbreak strain, and a good vaccine will most likely protect. But quality of the vaccine is still an issue. There are not vaccine producers in Africa that have proven WUA required potency. High quality vaccines can also protect against infection when the strains are antigenically significantly different, R1 value below zero point 0.3, showing that vaccine quality is important. Therefore, it is important to do post-vaccination monitoring according to the FAO or WUA guidelines. I don't know if you want to
Oh.
comment on that.
Yeah, sure. Aldo, thank you so much for this. Maybe I must have tweaked something. Yeah, but thank you so much for this. Absolutely. So that means that if there is no significant difference between the vaccine strain and the outbreak strain, as you well put it, it's most likely to protect. But if it's less than zero point 0.3, that means that there's quite a significant difference, and therefore the vaccine strain may not be able to protect against the circulating strains of the virus. Sorry, could have been some kind of tweak in
Yeah.
I was going through. Thank you for that, Aldo.
And then just a follow up, can you tell us how the post vaccination monitoring is done as well?
Yes, like I explained before, that we've had very few studies that have been carried out on vaccine monitoring. Actually, I think the study that I did together with some colleagues from Purbright was one of the first post-vaccination monitoring studies that we did. So we followed the post-vaccination monitoring guidelines that were released by the FAO. and it was in a controlled study. But basically, we did it actually at my institute. Controlled study. The article is actually out. If you look for Kerfua et al. 2024, it's out there, and it can give you details also of actually what we did. Now we are planning, because most recently Uganda had quite a lot of outbreaks that were registered in over half of the districts in the country. And we had some kind of policy change, and the minister was like, okay, we have to go out there and do a massive vaccination. So we want to go out there and do some post-vaccination monitoring to find out, how well did these vaccines actually work? Did they work the way they were supposed to? And then give feedback to the
Mm.
to the vaccine producers, to the industry, and tell them this vaccine did not do as you had said it would. So maybe there is some work you need to go back and do about it. So
Mhm.
Alda, that's a nutshell of it. But kindly, you can look for our publication, Kerfua et al. 2024. Thank you.
Thanks, Susan. So next question it's from Alice Mulondo. Thank you for that brilliant presentation, Dr. Kafua. Why do you think, or what do you think contributed to the disappearance of serotype C in Kenya? Do you know if there are any studies going on right now to determine that disappearance?
Thank you, Dr. Alice. Now, this is not an easy question. I think this is what the World Reference Lab for Foot and Mouth Disease is still trying to answer. First of all, they're trying to answer: has it really truly disappeared, or is it that our surveillance systems or programs are not capturing this? Um, Given that we know that in Africa, especially, let me talk about East Africa, where I'm from, we don't have rigorous surveillance systems. So is it that maybe we could be missing out on some of these samples? I don't know. And also the fact that if we look at the diagnostic tools now, most of the labs
Mm.
are actually not including C, and not including type C, serotype C rather, in their diagnosis. So maybe it's something we have to be very deliberate about. That's my thought. That we could be deliberate about this. Like if we followed up every single outbreak and tested it, could we find it? So I don't have a very good answer to this, but I think the World Reference Lab is really trying to see if really it has disappeared, or if we are just not doing good surveillance.
Thanks. Next question, Nicholas Denomandi. Apologies if I'm not pronouncing your name correctly. Question is, why don't you consider, as does WRL, including in the vaccine matching the heterologous VNP rather than only R1, since the latter is a ratio and therefore can mislead the interpretation?
Yeah, that is very true, Nicholas. Thanks for that question. Yes, for now we are still trying to get all this data together. We've not yet done anything conclusive. So for now we are just looking at the homologous BNT. Maybe later on we shall consider the heterologous one and then compare and then see what we can deduce from that. Thank you.
Okay. Next question from Sinan Akhtas. Why do you what do you think about evaluation of vaccine strains using East African antigen panel?
Yes, yes, that's a good question because even for our study, we actually did our evaluation basing on the East African antigen panel, and the results are displayed there, and they were quite interesting. So I just sign on. I just ask you to maybe look through our publication. We actually did this, and I think it's a good breakthrough because we need to know what is the standard. I mean, otherwise we wouldn't test this against all kinds of different strains. So I think this is a very good question. We did this for our vaccine evaluation study. Kindly can look through our article. Thank you.
Okay, another question from Nicholas. You say vaccines in Kenya are matching, but none of the vaccines in the WRL list are yet used in Kenya. Any comment on that?
Yes, I do have a comment on that. I think what the WRL does is to just test, do a vaccine match against the circulating strains, maybe in order to give recommendations for use. I think this is a good practice because it helps a country to know, what can I now use, what can I use for for this particular circulating strain. So well, Nicholas, I know works in the vaccine industry. So if he says that, then I guess it's right. But I think it's an important practice to do vaccine matching in order to make good recommendations. And now that we have the East African panel, I mean, all the better for us, all the better for us. But it's a good practice that we should be able to do. Thank
Okay,
you,
thank
Nicholas.
you.
Mhm.
Next question from Benedicto. Based on your experience, what do you think are the most likely reasons why suspected FMD samples collected and submitted for testing often return negative results? And what would you recommend as the main solution to address this challenge, given that emergency vaccination is the primary strategy in countries like Uganda? How does the need for a vaccine that matches the outbreak strain affect the effectiveness of this approach? Yeah.
Oh, thanks, Benedicto. These are like how many questions? Three.
Yep.
Um. Okay, I'll just read this. What do you think are the most likely reasons why suspected MMV samples collected often run negative results? Oh, well, like I displayed in the reflections. Those are just reflections. Could it be that there was issues with the cold chain? Could it have been issues with the way the sample was collected? Was the right sample collected? Considering the FMDV window period, did the people who went out there, uh, did they go out and collect the samples in the time in which the virus would be able to would be found in the material? So all those are things that we have to think about, we have to discuss. So there may be even others that I have not mentioned. So this brings in the issue of training. People need to be trained, given refresher courses, and I'm very grateful to the EUFMD that has this training. So I think labs that are working on foot and mouth disease research should be able to make use of these trainings, and maybe we need to do some kind of evaluation. Like we put up an intervention, like say training and intensive training, and then we see if there's a difference in in the quality of samples that are gotten. And then, what would you recommend as the main solution to address this challenge? Yeah, I've already said it. We need to do training and figure out if the trainings are very helpful. Given that emergency vaccination is a priority, how does it for vaccine emergency? Yeah, I think I've explained this, and also we had a notification from the some people asked questions that it would be better to do the heterologous vaccine matching. Yeah, so the vaccine matching helps a lot to let you know, like to give recommendations, possible recommendations of which vaccine strains can be constituted in a vaccine, and that may be protective. So that's how it works. If you get a vaccine strain that has that is close to the circulating virus, it may mean that that vaccine strain could be protective against that circulating virus. So you could actually include that vaccine strain for administration, for vaccine administration for control. Yes. Thank you, Benedicto.
Okay, thanks, Susan. Question from Sanan Akhtas, again questioning post-vaccination surveillance to evaluate the success of vaccination campaigns. How is that done? And how do these countries maintain cold chain in order to make sure that the quality of the vaccine is good throughout the process?
Sorry, I didn't get that.
So
No.
just, with the post-vaccination surveillance and how is when the vaccines are administered, is there a cold chain being maintained through that vaccination process
Oh
to
yes, thank you,
ensure
Sina.
the quality of the vaccine? Yeah.
Yes, thank you, Sinan. Yes, this is a very good question because you find that sometimes the vaccinations have to be carried out in the rural areas of the country. It is something that the ministry has taken charge of, and they have ensured to supply at least every laboratory. in the districts with refrigerators that are able to maintain this cold chain. But of course, we know that there are usually some challenges, and that is why we want to conduct the post-vaccination uh, monitoring for these recent vaccination, exercises that have been carried out, in order to ask some of those questions. Because you may find that people go out, the veterinarians go out there and spend a longer time than they would have thought, and probably not carried sufficient ice. So how can we be able to bridge those gaps? If you're going to a faraway place, how can we be able to support that? How can the ministry be able to support that? So those are very good questions that we hope we shall be able to get the answers to after we've done this post-vaccination monitoring. Yeah, thank you very much.
Thanks, Susan. Next question from Mary Lou Baninga. Would wildlife play a role in the temporal spatial spread of serotypes or topotypes? Has any wildlife studies been done to determine their role in FMD outbreaks?
Yes, yes, absolutely. I think so. Anyway, usually this wildlife and livestock thing has a lot of contention. Personally, I've not done a lot of work on wildlife surveillance of foot and mouth disease, but I think there must be something. Because if we look at some of the studies that have been done, especially in Uganda, there are not so many though. In maybe about published studies, maybe about three published studies from Uganda on wildlife, three or four published studies. When you look at Kenya, there was actually a most recent study that was carried out. That was, I think, in 2022. That was carried out, and also previous studies by Sabenzia and some other scientists. Um, there was no substantive conclusion on if wildlife was able to pass on the virus to the livestock. But we know that yes, wildlife can be carriers of the virus, and can be able to play a role in the epidemiology of the disease. So I can't give anything very conclusive for now, because there are people actually doing these studies. I think also Melanie, you're one of the people doing these studies. So I think there's still more information that will come out on this.
Yes, definitely. I think we have time for maybe one more question, and we'll take the question from Richard Sesyanzi. Sorry, I can't say your surname. Thank you, Dr. Susan, for this presentation. In this presentation, it's clear the various serotypes do prevail. I don't know, I might have missed it, but out of these serotypes, which one is the more virulent, and how does it differ from other strains? Have you done some polymorphism like SNPs analysis or other evolution analysis?
Yeah, thank you. That's a very good question. Uh, from the experience in the field, if you talk to the farmers, they'll tell you, this time this foot and mouth disease really hit us.
Mm.
They'll tell you animals died. They'll tell you the signs were so, they were so pronounced. They'll tell you it was different. They'll just let you know it was different. And those are some of the things that I think we need to take note of. There's a project that I'm working on, and we're trying to note all that down. Like, if there's an outbreak, what are some of the signs that are exhibited? Because I think it is true that the serotypes will exhibit different signs. So those are some of the things that we need to note in the field setting. I'm sure that people have done a lot of work in the lab setting, in a controlled setting, but what do we see out there in the field? So thanks, Richard. These are some of the questions that are going to be answered probably by some of the studies that we're doing right now.
Okay, we have unfortunately run out of time. There are some questions that are still unanswered. We will try and submit answers to them. We will send that to Susan to answer that for us, and we will try and distribute the answers to that. And just a reminder to all participants as well that we do post these webinars on our YouTube channel. So please take the time to have a look at it there. Even if you do miss the live webinars, you will find it on the YouTube link for the GFRA. And you are welcome to post questions there. And we will monitor the questions for a month after a live webinar has been completed. And we will try and get the answers to you also from there. So we try to accommodate all the time zones, but it's not possible always. So I know some of our colleagues are missing the live webinars. So we are trying to accommodate this through the YouTube link. So please do feel free to go on there, and you will see all of our past webinars there as well. So Susan, thank you very much. It was really a very nice presentation, very nice conversations had. And I think I also have a few questions that I would like to pose to you as well. And I think some of our participants will also do the same. So thank you for your time. Thank you for being our first GFRA webinar presenter. We really appreciate it. And thank you, everyone. I'm going to hand you over to Kayla now, who will wrap up for us.
Yes, thank you both, and thank you to everyone who joined today. As you exit the webinar, you will see a survey pop up, just a quick survey. If you could just take a minute or two to fill that out. We really appreciate that, and we really do use those surveys to continue to improve on this webinar series for the GFRA. All right, thank you all. Have a great rest of your day.
Thank you. Goodbye.
Thank you.